Hi everyone, welcome to the last 6 or so chapters of Bio 181, now available for you online. I'll be recording lectures for you that will go with each chapter with the slides. Just like before, the slides are going to be available for you to look at online, and you'll be hearing me right here through these videos. Also keep in mind that I wasn't planning on recording these things at home, so I have to apologize if there's the occasional door bark or dog bark or doorbell or 4-year-old singing something from Frozen 2. At the same time, these chapters, you'll be able to watch them at 2 a.m. or whenever it is that is most convenient for you. Also keep in mind that these aren't live, so you won't be able to ask me your questions in real time. However, please do email them to me because other students might have the same questions. I'll be sure to post the response to any questions on the just on the announcement page, but I won't include your name. So again, if you have a question or if something isn't clear, certainly go ahead and email me so I can get that out to you. Also, just like in class, these chapters, they will be helpful compared to just reading the book in that I will absolutely highlight material that I'm going to ask you about, as we'll still have tests. I'll also alert you to material that is available for you to learn about, but that you won't necessarily be asked about on class or on exams. And so, our first of our online chapters, Chapter 10. In Chapter 10, we're going to be looking at chromosomes, mitosis, and meiosis. And this all comes back to deoxyribonucleic acid, or DNA. So all cells contain DNA, and that's coded genetic information. Genes within DNA control a cell's activities, and these genes are passed on to descendants. When a cell divides, DNA is replicated and copies are transmitted to each daughter cell. Now, each DNA molecule is packaged with proteins to form a chromosome. There are a couple of different types of processes where we'll see DNA replicate and split, but how it does this depends on what the end goal is. When we need to replicate a cell and make it identical to the parent cell, so this would be maybe in a growing organism where we just need more liver cells or more muscle cells, or if we're replacing some cells, such as red blood cells, we're going to undergo mitosis. In mitosis, a parent cell transmits one copy of every chromosome to each of its two daughter cells. So in the end, we wind up having one parent cell become two daughter cells. In meiosis, this is where organisms that reproduce sexually, such as humans or dogs, in this process, the chromosome number is reduced in half to produce eggs and sperm. Then the original number is restored in the zygote, so that egg and the sperm come together and we get a zygote, and now we're back up to that organism's normal chromosome number. We'll be going through both mitosis and meiosis in much greater detail. We're going to be focusing our talk on eukaryotic organisms, in part because we don't see bacteria undergoing sexual reproduction, so meiosis just isn't an option there. And also, the chromosomes between eukaryotic organisms and bacteria, or most of your prokaryotic organisms, are a bit different. We will talk about those prokaryotes at the end, but again, our focus is going to be on eukaryotic organisms, like you. So here you can see human chromosomes forming from an unidentified cell, and these are being looked at with fluorescent light microscopy. We'll be going through some of the characteristics of a chromosome in just a minute and looking at those guys in greater detail. Now, when a cell is not dividing, so when your cell is just sitting there doing its regular work, the chromosomes are all present in an extended, partially unraveled form. And this allows for us to have some enzymes go ahead and access the genes that that cell needs to produce on a regular basis. Now, when cell division is occurring, the chromatin fibers are going to really condense and the chromosomes become visible as distinct structures. And so when we're seeing images like this with these well-defined chromosomes, we know that this cell is undergoing cell division. So if we had all of our DNA unraveled all of the time, it would be insanely long. It's several feet long, and then we're trying to cram that into each and every single cell. And also, not every cell needs to access all of its DNA. Remember that the DNA in your liver cells is going to be the same coded DNA as in the cells in your retina in your eye or in the muscles in your tongue, even though those organs and those tissues have very different functions. It all has the information, but a lot of it just kind of keeps in its back pocket. It's there if some lab wants to use it, but the cell itself doesn't really need to use a lot of that. And so a good chunk of DNA is kind of wrapped up around these proteins called histones. And these wind up forming little nucleosomes. And so here you can see a model for the structure of a nucleosome. Each nucleosome bead contains a set of 8 histone molecules forming a protein core around which the double-stranded DNA can wind. The DNA surrounding the histone consists of about 146 nucleotide pairs. And then there's another segment of DNA, about 60 nucleotide pairs long, that links these nucleosome beads. And so wrapping that DNA that we don't need to access right then around these histones to form nucleosomes helps us to kind of condense that DNA a little bit. Here you can see an electron microscope image of those nucleosomes. You can see they almost look like a string of pearls. Those short 60 nucleotide segments in between each of those little histone sets with the DNA wrapped around them. So as I mentioned before, histones are an important part of the regulation of gene expression, whether genes are turned off or on. Now remember that in all the cells of your body, you have the same DNA. But in some cells, some of your DNA is going to be relatively inaccessible. Those genes will be turned off. In other areas, particular genes are going to be turned on. Those cells are going to need to make those genes or make the proteins that are produced by those genes. When genes are turned on, we'll have the DNA being released from the histone proteins, which you can see here. You can notice all of those DNA loops, but even still, notice how densely packed the DNA strands are, even though they've been released from the histone proteins that organize them into tightly coiled structures. Remember that during cell replication, having all of those loose strands of DNA would not be very helpful, so we make sure that that DNA is as tightly packaged as possible. That's when we'll see it in its most organized structure. And also, while the cell is replicating, it's not really going about doing much of the other work. It's focused on its primary job of replicating during that moment. So here we can see how DNA is packaged into highly condensed metaphase chromosomes. Again, that's when we're going to see that DNA most condensed. So first DNA is wrapped around histone proteins to form nucleosomes. Those nucleosomes are about 10 nanometers in diameter. Then in the picture farthest to the right, you can see the histone H1 associates with the linker DNA, packaging adjacent nucleosomes together into a 30 nanometer chromatin fiber. So you can see again how they're kind of coiling around themselves. Then those chromatin fibers form large coiled loops that are held together by scaffolding proteins. And then condensin is going to bind to DNA and wrap that into coiled loops that are compacted into the chromosome. And so we have this kind of continuously wrapping structure. So we'll take that thin strand of DNA, wrap it around those histones, then we wrap those around themselves, then we'll wrap that fiber into coils, and then we'll wrap those coils around themselves until we get to that super condensed chromosome. So every species has a characteristic number of chromosomes in the nucleus of each somatic cell. A somatic cell is going to be most of the cells in an organism. So for you, cells in your heart, your brain, your kidney, those guys are all somatic cells. The other cells are the reproductive cells. So those are going to be the ones such as eggs and sperm. And so humans, they have 46 chromosomes. Dogs, 78 chromosomes. But roundworms, they've got a whole 2 chromosomes. Now some ferns have more than 1,000 chromosomes. It's the information specified in the genes that makes each species unique. The number of chromosomes does not correlate to the complexity or intelligence of a species. Again, humans have 46, and some ferns have more than 1,000 chromosomes. And every single one of you is brighter than a fern. So when cells reach a certain size, they usually either stop growing or they divide. And so there's a cell cycle. Those are the stages through which a cell passes from one division to the next. Timing varies. It's usually 8 to 20 hours in actively growing plant cells, and it consists of two main phases, interphase and M phase. We're going to spend a little bit of time talking about interphase, and then we'll spend a lot of time talking about M phase, because M phase includes mitosis and cytokinesis. Now, most of a cell's life is spent in interphase. That's the time when no actual cell division is occurring. The cell cycle, which is again a successive series of events in the life of a cell, includes interphase, which has G1, S, and G2, and then M phase, which includes mitosis and cytokinesis. Proportion amounts of time spent at each stage varies among species, cell types, and the growth conditions. But if we had a cell cycle that had a period of about 12 hours, G1 would take about 5 hours, S phase would take about 4.5 hours, G2 would be about 2 hours, and then M phase would be 30 minutes. And again, we'll be spending the most time though talking about M phase because that's where a lot of these different steps occur. So let's take some time to talk about interphase. And again, that's where cells are most of the time. During interphase, the cell is going to carry out normal life activities, and the chromosomes, though, also become duplicated. And so during interphase, the cell's just still doing its normal stuff. However, we're also seeing duplication of the chromosomes. There are 3 phases of interphase. So in G1 phase, we'll have growth and normal metabolism. And this is going to be between the end of mitosis and the beginning of the S phase. And so during this time, the cell is doing its normal things. We'll have proteins made for export. Oh, we need to make some more mitochondria. Okay, and just doing all those things, and it's growing. Now, S phase, this is when DNA replicates and those histone proteins are synthesized. And so again, S phase stands for synthesis phase. We're replicating DNA. We're synthesizing the DNA. After S phase, we'll have the G2 phase. Here we'll see protein synthesis increase in preparation for division. Because remember, when that cell divides, we're also going to need to send along all the appropriate proteins, now not just for one cell, but for 2 cells. So M phase includes 2 main processes, mitosis and cytokinesis. Mitosis is a nuclear division that produces 2 nuclei containing chromosomes identical to the parent nucleus. So mitosis is going to be that division of the nucleus. Mitosis has 5 stages. In some texts, or if you use something like the Khan Academy, or if you're finding some other reference materials, you might see 4 stages. But prophase, prometaphase, metaphase, anaphase, and telophase are those 5 stages. In some things, you might see prometaphase, which is between prophase and metaphase, not included. Our text does include prometaphase, and we're going to go into each of these steps in greater detail. Now, after mitosis, we've got, again, 2 nuclei, but we're still in one big cell. So cytokinesis is going to be the division of that cell cytoplasm to form 2 cells. And so to help you remember the order in which these steps are occurring, I like the mnemonic PP-MAT. And so, yep, look at that little Frenchie wanting to go pee on that PP-MAT. And so prophase, prometaphase, metaphase, anaphase, telophase. PP-MAT, like this little Frenchie getting ready to pee inside your house. Let's start talking about metaphase. So during metaphase, we'll see that begin with chromosome compaction. And so we had our DNA synthesized or replicated during the S phase of interphase. And now we have these duplicated chromosomes that consist of identical sister chromatids. And so if you look at the picture on the left, you see that this particular organism has 4 chromosomes. We're going to assume that the red ones are maternal, that the organism inherited them from the mother's egg. And the blue ones are paternal. The organism inherited them from the father's sperm. Now, we always have pairs of chromosomes. And so we have 23 pairs of chromosomes for a total of 46 chromosomes. This organism has 4 chromosomes or 2 pairs. The big ones on the left are a pair of chromosomes, and then the smaller ones on the right are a pair of chromosomes. Now, during the S phase, each of those chromosomes wound up getting replicated, but they're still kind of connected together. And so you'll see that they kind of form like this little X. They're these little blue bubbles kind of right in the middle. But each of those strands is going to be a sister chromatid. And those two sister chromatids should be identical to one another. Those sister chromatids are held together at the centromere, those little blue dots in the middle, by cohesins. And then attached to each centromere is a kinetochore to which microtubules can bind. And we're going to see the function of that in just a second. Now, each pole of the cell contains a microtubule organizing center from which the microtubules extend. Now, animal cells, we have a pair of centrioles in the middle of each microtubule organizing center surrounded by pericentriolar material. And then these microtubules are going to radiate from the pericentriolar material towards the chromosomes, forming the mitotic spindle. So you can see that in the image again, that those little yellowish kind of cylinders on each of the I guess the west pole and the east pole are the centrioles, and then you can see these fibers kind of going out between them, and those are the developing mitotic spindle. So language and vocabulary is really important in this section because we have a lot of words that sound similar: chromosomes, chromatids, chromatin. What you're seeing here pictured is one chromosome, or rather an electron microscope image of a chromosome and then a drawing that is similar to that. And so we've got a chromosome pictured here. This particular chromosome is currently made up of two identical sister chromatids. That chromosome made a copy of itself during the S phase of interphase, and these sister chromatids are two identical copies of each other, and they are tightly joined at that centromere region, that little pinched in spot. Later on during the process of mitosis, these sister chromatids are going to be pulled apart and will wind up with one copy of each, or one of each, in each of the cells that we're going to make. And so I strongly recommend that you make flashcards, use the flashcards that are in Cengage regarding all of these words that begin with chrome, because we're going to be seeing quite a few of them, and they're all pretty similar. So here you can see again the sister chromatids, each consisting of tightly coiled chromatin fibers. They're tightly associated at the centromere regions, indicated by those little brackets there. And associated with each centromere is a kinetochore, which serves as a microtubule attachment site. And kinetochores and microtubules are not visible in the transmission electron microscope image of this metaphase chromosome. So you don't see those microtubules or the kinetochore, but they are there. Prometaphase is next, and this is not a stage that you might see listed in some older text or in some materials. So again, if you're looking at like a Khan Academy or some other additional resource, they may just go from prophase to metaphase. But the text we're using does consider prometaphase its own separate phase. And so prometaphase begins when the nuclear envelope breaks down. The spindle microtubules, they'll shorten or lengthen as they wind up contacting chromosomes. So you can see in the picture on the right that some of those microtubules are coming into contact with the kinetochores, and then they're adjusting their length. Now, one of the kinetochores of a duplicated chromosome winds up capturing a microtubule, and then the chromosomes start moving towards the cell's midplane. So you can see currently they're all scattered about, but soon we're going to line them up along like a straight line going down the middle. Now the cohesins, they wind up dissociating from the sister chromatid arms, helping them to free from one another because we are going to need those to pull apart. And so again, in prometaphase, the big things that we see are that the nuclear envelope has broken down or is in the process of breaking down. The spindle microtubules attach to the kinetochores of the chromosomes, and the chromosomes begin to move towards the cell's midplane. Metaphase is maybe one of the most commonly recognized images during mitosis. And so during metaphase, all of the cell's chromosomes are aligned at the cell's metaphase plate. So just this center line going down the middle. Here it's drawn so that the line is vertical, but you could also have those centriole pairs being at the north and south pole and have the metaphase plate going across there horizontally. Big thing is that there's a line going down the middle, and all of the chromosomes are lined up along the middle, and there's a microtubule attached to each kinetochore. Now, during metaphase, each chromatid is completely condensed, and so this is as tight as they're going to get. The mitotic spindle has 3 types of microtubules, and the remaining cohesins that were kind of joining those two sister chromatids, they dissociate. Now here we'll often kind of capture in a lab the cells during metaphase because chromosome abnormalities can be detected using metaphase chromosomes to construct a karyotype. And we'll be seeing karyotypes in a little bit. And so again, metaphase, big things are that the chromosomes line up along the cell's midplane, and the spindle microtubules attach to each chromosome or attach each chromosome to both poles. We can take a closer look at the mitotic spindle here. And so on one end of each microtubule of this animal cell is associated with one of the poles. These astral microtubules, which here are represented in a green color, they radiate in all directions, forming the aster. Then we have kinetochore microtubules, which here are pictured in red, and they're going to connect the kinetochores to the pole. Then we also have polar, so non-kinetochore microtubules, which are in blue here, and they overlap at the midplane. And they'll all have roles while these guys are separating, and when we see separation start in anaphase. So anaphase is our next step, and this begins as sister chromatids separate into separate chromosomes that move towards opposite poles. The kinetochore microtubules shorten at their plus ends and pull chromosomes toward the poles. And this anaphase ends when all the chromosomes have reached the poles. And so here you can see that our previously paired sister chromatids have now separated and they're each their own chromosome. While they were together, they were one chromosome. So 2 sister chromatids can form one chromosome while they are attached at that centromere. However, once they're pulled apart during anaphase, we are now briefly looking at 2 chromosomes there. And so again, sister chromatids are going to separate at their centromeres, and one group of chromosomes moves towards each pole of the cell. And those spindle poles, you can see that that cell is now no longer as perfectly round as it was. It's kind of starting to pull apart and move, those poles are moving farther apart. Let's go ahead and take a closer look at those cohesins we've been talking about. And so when we have those sister chromatids joined together, we'll see that we've got cohesins keeping them together all throughout both of those molecules. And then we've got quite a few of them at that centromere region, which is why we see such a close association there. But as mitosis is progressing, some of those cohesins wind up dissociating from the duplicated chromosomes. And so they had less and less of a connection. But we still maintain it in that centromere region. However, once we get to anaphase, once all of our chromosomes are lined up along that midplane, and we've checked off that we have completed metaphase appropriately, then those centromere cohesins will dissociate, and then we can have those daughter chromosomes separate during anaphase. And so there have also been experiments done to determine how chromosomes are transported towards the spindle poles during anaphase. And so what they did was they used photo bleaching here. And so they marked the microtubules using a little laser microbeam that wound up bleaching the dye but keeping the microtubules intact. So we grabbed these guys. You can see that they're lined up. It looks like metaphase was just ended and maybe we're about to start anaphase. And then they let those sister chromatids get pulled apart. And so what they saw was that the chromosomes were moving towards the bleached end. And so the areas that were getting shorter were closer to the chromosomes, not to the centrioles. And so this told them that the microtubules were being disassembled at the kinetochore end, not at the centriole end. So telophase is our last stage of mitosis. And so in telophase, the chromosomes arrive at the poles and they return to their interphase-like conditions. So they start to spread out a little bit. We're not as tightly condensed. And they decondense by partially uncoiling. A new nuclear envelope forms around each set of chromosomes, which you can see more clearly in the picture on the right, that we're starting to get those nuclear envelope fragments back together again. And spindle microtubules, they disappear and the nucleoli reorganize. And so we don't see those long microtubules coming out of the centrioles anymore. Now note that we are still kind of a single cell, although in the middle we're starting to form what we call a cleavage furrow, and we'll see a little bit more of that in the next slide. But the cell's starting to pinch off a little bit, but still technically is one giant cell about to become two. And so again, here we've got chromosomes are finally grouped at the poles. The chromosomes decondense and the nuclear envelopes begin to form. And then our final step after mitosis, cytokinesis, is going to result in two daughter cells. So cytokinesis is that last stage. It's not officially part of mitosis. Our chromosomes have separated and we're not worrying about the nucleus anymore, but now we're just dealing with the rest of the cell. And so cytokinesis is going to be the division of the cytoplasm to yield two daughter cells. And this is generally going to begin during telophase and overlaps mitosis a little bit, because once those chromosomes are at each pole, we can start, as you saw in the previous slide, pinching in a little bit in the middle. And so in animal and fungal cells, an actomyosin contractile ring attaches to the plasma membrane and causes constriction. And so you can see it kind of pinching off there in the middle. And that cleavage furrow deepens and then ultimately separates that cytoplasm. But remember that the cell membrane is made up of that phospholipid bilayer, and so it's easy for that to kind of fill in that space as it is kind of budding off there. You can see in this image here that this is a transmission electron microscope of the equatorial region of a cultured animal cell undergoing cytokinesis, but you can see that cleavage furrow pretty clearly. Dividing fungal cells will also have that contractile ring that causes cytokinesis. Now, each of these two daughter cells receives exactly the same number and the same kinds of chromosomes of parent cells, so the nuclei should be identical. Now, organelles are apportioned with cytoplasm that each daughter cell receives during cytokinesis. So if there were, 100 mitochondria, each one gets about 50. Nobody cares if one gets 49 and the other one gets 51, and close enough. The mitochondria and the chloroplasts contain their own DNA, and they form by division of previously existing mitochondria or plastids. And we won't really be covering that much in this class at all. And so you might be asking yourself, what about plants? They have a cell wall. Yeah, that's right. So how are we going to do that? We can't just pinch off and kind of bud there. And so plants, they do something a little bit different. During telophase, we'll start to see that these vesicles gather on the cell's midplane. And these vesicles, they start to fuse, forming some larger vesicles. And then eventually there's one giant vesicle going all the way across. And that ultimately becomes a new cell wall from those vesicle contents. And so here we don't really see that same cleavage furrow form. We see those vesicles line up and start to separate the two daughter cells, and then those vesicles become a wall. And think about that in terms of when you were looking at like the onion cell and thinking about how lined up all of those cells were. Now that you know how they divide, reconsider why they were lined up like that when you were looking at them in the onion cell. So we've been talking about animal cells and fungal cells, and then a little bit about plants. They all have their separate chromosomes. Remember, roundworms with their 2, some ferns having thousands. But what about bacteria? So bacteria are a little bit different. They have this single round strand of DNA. So their bacterial chromosome is circular. It's a ring. And so they do not replicate in the same way that we do. What happens there is they undergo a process called binary fission. So binary fission is a precisely orchestrated sequence of events that ensures that each bacterial daughter cell has identical genetic material. And the bacterial chromosome is much longer than is depicted here in this picture, and it's tethered to the plasma at one spot, which we also don't see here. And so in binary fission, DNA replication begins at a single site on the bacterial chromosome, and replication continues as replication enzymes work in both directions from the site where replication began. And then once replication is completed and the daughter chromosomes separate, we see what's called a Z-ring forming at the cell's midsection. And the cell begins to divide as plasma membrane grows inward. And then a new cell wall forms and binary fission is complete, and we've got two identical daughter cells as a result. With that circular DNA, we and just one big old ring of DNA, we don't need to worry about making sure that one of each chromosome gets into each cell. There's going to be two circles of DNA at the end of their replication, and so we need to put one circle in each of those two cells. As you can imagine, things can go wrong during this process. Maybe we don't need the cell to make another copy of itself, like that area is all full, we don't need any more cells. Maybe the DNA isn't copied properly, or maybe the chromosomes don't line up correctly. And so we've got a lot of different checkpoints in this cycle. And so under optimal conditions, the length of the eukaryotic cell cycle is constant for a given cell type. And we've got cell cycle checkpoints that ensure all events of a particular stage have been completed before the next stage begins. This is kind of similar to making sure that you've got a passing grade in Bio 181 before you go on to Bio 182. If a gene that encodes a molecule involved in the checkpoints is defective, cancer or some other serious diseases can result. And so here we see some checkpoints in that cell cycle that we saw earlier. So the cell cycles consist of hundreds of sequential events, and those red bars show 3 important checkpoints that determine the previous steps are completed so the next steps may proceed. Each checkpoint is inactivated after it has performed its function, allowing the cell cycle to continue. And so that first one, the G1S checkpoint, this first checkpoint ensures that the cell has the necessary growth factors, nutrients, and enzymes to synthesize DNA. Without the proper signals that the cell is ready to proceed, the checkpoint will not let DNA synthesis begin, because there's no point in having 2 copies of that DNA if we don't need to have it. Don't use those resources to start that synthesis if we don't need to make more of those cells. And so growth factor is probably one of the most important things. And so the body will secrete growth factor when it needs more of a particular type of cell. Growth factor is also one way that cancer cells can grow out of control. And so sometimes you'll have tumor cells that either secrete their own growth factor or wind up basically forcing the constant secretion of growth factor by that organism. And so growth factor is one way that cancer cells kind of get around things. Now the next one, if you pass that G1S checkpoint, you're going to go ahead and synthesize that DNA. Then you're going to make sure that you've got all the other materials ready during G2. But then we get to that G2M checkpoint. And so that cell cycle checkpoint ensures that DNA replication is finished before the cell begins mitosis, right? There's no point in starting to divide the cell if I've only copied 22 of the 23 chromosomes. I need all 23 pairs copied. If a cell has damaged or unreplicated DNA, the checkpoint is not going to let the cell undergo mitosis. We don't want to go and make 2 copies of a cell that isn't correct. And then during mitosis, there's a metaphase anaphase checkpoint. And so remember, during metaphase, all of the chromosomes line up along that metaphase plate, that midline plate. And so when they're all lined up, we should have the chromosomes lined up and a sister chromatid on each side. And so I want to make sure that all of those chromosomes are lined up and that I've got a microtubule connected to each kinetochore, because if I have one that isn't lined up or isn't connected to a kinetochore, when they start splitting apart, all of a sudden I might not get one of those sister chromatids in each of my new cells. And so the metaphase anaphase checkpoint is sometimes called the spindle checkpoint. And this checkpoint occurs at the end of metaphase and prevents anaphase from occurring until all kinetochores are properly attached to the spindles along the cell's midplane. So far we've been talking about mitosis, or for bacteria or prokaryotic organisms, binary fission. And that is going to happen in somatic cells. And so again, your non-reproductive cells. Those cells are going to go and make identical copies of one another. What we're going to talk about next is meiosis, which is used in sexual reproduction. And so this is going to only occur in those cells that are forming eggs or sperm that are going to be used for sexual reproduction. Now remember that you have 23 pairs of chromosomes. And so To make sure that you always have 23 pairs of chromosomes, what happens during meiosis is that we wind up making a gamete, so an egg or a sperm, that has half of that number. And it's going to be a very specific half. We're going to make sure that we have one of each pair. So your eggs and your sperm have 23 chromosomes, whereas the rest of the cells in your body have 23 pairs of chromosomes. And so we're going to be looking again at sexual reproduction and meiosis for the next bit. Now, asexual reproduction is what we see in bacteria, and that's where a single parent splits to produce a clone. It's rapid and efficient. It favors organisms that are well adapted to their environment. There are ways that they can make some changes with things like plasmids, and we won't really be getting into that in this course much at all. We're going to be spending a fair bit of time, in fact, the next several lectures, talking about sexual reproduction and about genes and about just passing on particular traits. And so this is going to involve the union of an egg and a sperm to form a zygote. It also results in a lot of genetic variation among offspring. For those of you with siblings, unless you are an identical twin, you do not have the same exact DNA as your siblings. You have a lot in common. You have a lot in common with both your parents and with all of your siblings, but you're not identical. Sexual reproduction also favors adaptation to changing environments, and we're going to be looking at that towards the very end of the class, but also a little bit kind of in the next sections, but really focusing on that at the end. To understand meiosis, it's first helpful to understand homologous chromosomes. So remember that you get one chromosome from your mom and one chromosome from your dad. And both of them are going to have information about eye color, hair color, making red blood cells, or whatever is on that particular chromosome. But there might be slight differences. Again, perhaps your mom passed on a gene for brown eyes, but your dad passed on a gene for blue eyes. Still going to have eye color, but we've got two copies of this piece of information. And so these homologous chromosomes, these are paired chromosomes in somatic cells. You can see here that they are similar in shape, size, and position of their centromeres. They carry information about the same genetic traits, but it's not always the same information. We'll give names to those things in the next week or so. So human cells have 46 chromosomes in 23 homologous pairs. Now you'll notice that the first 22 of those pictured here, they're all pretty similar in size and shape. And then you see that last one, 23, is actually a little bit different. So those are our sex chromosomes, and those are going to be either XX in female, and you can see here that the XX, similar, or we can have X and Y for male. The Y chromosome is definitely much smaller. It's kind of close to like one of those chromosomes like in size to like 21 or 22. And so they are not exactly going to be the same in size, shape, and position of their centromeres. So the XY can be different, but those first 22 you can see, they all match up pretty well. And so a set of chromosomes has one of each kind of chromosome, one from the maternal parent and one from the paternal parent. Now, when that's the case, we have a diploid chromosome number. And so that's a cell or nucleus containing two sets of chromosomes. So here you can see we've got 2 of chromosome number 1. We've got 2 of chromosome number 2. We've got 2 of chromosome number 3, and so on. And so this is the diploid set. And so we have 46 chromosomes here total. However, if when we are having offspring, if both parents passed on 46 chromosomes, all of a sudden our organism, the offspring, would have 92 chromosomes. And that's different. That's not right. We want to make sure that this maintains that same number. And so when we're making gametes, either sperm or eggs, we're going to wind up taking only one of the chromosomes from the homologous pairs and passing that on. In that case, we are looking at the haploid number, or a cell or a nucleus that only has a single set of chromosomes. And so in humans, the diploid chromosome number is 46 and the haploid number is 23. So here you can see that cyclical nature of maintaining the chromosome number. So gametes are haploid. We demonstrate that, or we indicate that with N. Remember, gametes for us are going to be eggs and sperm. Now, when an egg and sperm fuse at fertilization, we have N in sperm plus N in an egg, and that makes 2N, the zygote. And so each of those gametes contributes one set of chromosomes, and we have that diploid number being restored in the zygote, which is 2N. So then that zygote grows into whatever, an adorable baby, big person, and then they start making eggs or sperm. And when that zygote is dividing by mitosis, all of the somatic cells receive the same diploid set of chromosomes. Remember that mitosis is going to produce identical chromosomes or identical cells. And now, though, when that organism winds up going and making eggs or sperm, those guys will divide by meiosis, giving us gametes with an N, with a haploid number. Now, an individual whose cells have 3 or more sets of chromosomes is polyploid, and we'll be talking about those situations. And polyploidy is rare among animals, and in many cases can result in defects, but it's pretty common among plants, as many as 80% of all flowering plants are polyploid, and it's also an important mechanism in plant evolution. So meiosis produces haploid cells with unique gene combination. This is how we get some diversity in our population. And so DNA in a diploid cell is going to duplicate once, but then it undergoes 2 divisions, yielding 4 haploid nuclei. And so again, we have a cell, and it's going to go through that cell cycle. It's going to go through S phase. It's going to make a copy of its DNA. It's going to divide. It's going to seem pretty similar to mitosis, but we'll point out some differences. But then without undergoing duplication again, that cell will divide again, producing 4 haploid nuclei. Each haploid nucleus contains one member of each homologous pair. So they either get the maternal chromosome for chromosome number 5, or they get the paternal chromosome for chromosome number 5. For chromosome number 4, again, they'll either get the maternal or paternal. They're not necessarily, it is statistically possible that they would get all maternal or all paternal chromosomes, statistically possible, but it's not super likely. Most commonly, we're going to see a mix of some chromosomes being paternal or predominantly paternal and some being predominantly maternal. Each homologous chromosome pair is shuffled, so the resulting haploid cells each have a unique combination of genes. And so meiosis consists of 2 nuclear and cytoplasmic divisions. We call them meiosis 1 and meiosis 2. And unlike our photoreceptors in photosynthesis, they do go in order, meiosis 1 and then meiosis 2. Each of those stages, meiosis 1 and meiosis 2, includes prophase, metaphase, anaphase, and telophase stages. Now in meiosis 1, those partner homologous chromosomes are going to pair. So instead of just having those sister chromatids line up on that metaphase plate, we actually have all 4. We've got 2 homologous pairs, the maternal and the paternal, lining up on that metaphase plate. And then they separate and move into different nuclei. Then in meiosis 2, the sister chromatids separate from each other and they're distributed to 2 different nuclei. We'll be looking at these stages in more detail. And so here, though, you can see kind of an overview where the figure begins with a diploid cell with 4 unduplicated chromosomes. The chromosomes, you can see in the next stage, they replicate their DNA. So now we've got sister chromatids bound together at that centromere. And so 4 chromosomes, each with 2 sister chromatids. And they also have the homologous pairs labeled. So we've got 2 chromosomes, we've got 4 chromosomes and 2 different types, 2 separate pairs. We'll see that those wind up splitting so that we get one of chromosome 1 and one of chromosome 2 in each of those 2 cells. And then in the meiosis 2, we'll see those sister chromatids get pulled apart. So the stages again are similar. In meiosis 1, we go through prophase 1, metaphase 1, anaphase 1, telophase 1. In meiosis 2, we go through prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 1 includes synapsis and crossing over, which we'll cover in a little bit more detail in a couple more slides. And so remember that each duplicated chromosome consists of 2 sister chromatids linked by cohesins. Our homologous chromosomes, so remember chromosome 1 from your mom and from your dad, align side by side, which results in the association of 4 chromatids. And so we have our homologous chromosomes pair up so that now we've got we've got 2 pairs of sister chromatids, because remember the sister chromatids are identical, and your maternal and your paternal chromosomes for chromosome 1 are not identical. They'll have all the same information about the same genes, but there might be different alleles, different specific pieces of information there. And so these homologous chromosomes are going to align side by side, which results in association of 4 chromatids, and we call that a tetrad. We can then have a synaptomial complex forming along the length of those synapsed homologs. And we'll have enzymes that break and rejoin the DNA molecules, allowing a process called crossing over. And we're going to see that again. We're going to look at a couple of slides of that in the future, because that's really important for how we get some of this genetic variation. And so again, we've got interphase. During interphase, before meiosis, the DNA replicates. It's pretty similar to mitosis there. But then in prophase 1, one big difference is that we don't just have a chromosome that with the 2 sister chromatids line up. We now have the 4 chromatids of the 2 homologous chromosomes kind of pairing up. And they're going to synapse and exchange segments by crossing over. And then we also see the nuclear envelope breakdown. So in meiosis 1, our tetrads lined up along that metaphase plate, and then they were pulled apart with one of the chromosomes with the 2 sister chromatids going to one side, and then the other of that chromosome, chromosome number 1 or chromosome number 4, being pulled across with its 2 sister chromatids. Then we get to meiosis 2. Remember, there is no further replication going from meiosis 1 to meiosis 2. We immediately go from meiosis 1, from the end of telophase 1 and cytokinesis, into meiosis 2. You can see here in the cells on the left, we're getting set up for prophase 2. And so the chromosomes, they condense again, just with this brief period of interkinesis, but the DNA does not duplicate. You can also see that we do not have homologous chromosomes here. If you look at the chromosomes in the cell to the most left, you'll see that there's a pair, like the big ones. So those are 2 sister chromatids, and then there's the little one. Those are 2 separate chromosomes. However, we still both have the sister chromatids joined for that chromosome. You can also see that even though that big one is mostly blue, one of the sister chromatids wound up crossing over and has a little chunk of red. And then we can see that even though the little chromosome is mostly red, there was some crossing over, and one of those sister chromatids has a little bit of blue. We can see its partner in the other cell. So the paternal, the blue large chromosome, wound up going into the cell on the left, but the maternal wound up going into the cell on the right. But you can see that little fragment where we had some crossing over. And then you can see the opposite with a smaller one. And so in prophase 2, it's similar to mitotic prophase, but there's no pairing of homologous chromosomes and no crossing over. And so they just kind of line up. And this is going to seem much more similar to mitosis, in that we're going to now pull the sister chromatids apart. And so then we go from prophase 2 to metaphase 2. Our chromosomes line up on the midplanes of their cells in groups of 2. And so again, not 4. So now we just have one of each chromosome lining up on that metaphase plate. Meiosis 2 proceeds and to anaphase 2, and our sister chromatids separate, and the chromosomes move to opposite poles. In telophase 2, the nuclei form at opposite poles of each end, and cytokinesis occurs. And in the end, we have 4 haploid cells. And so we have 4 gametes in the, for animals, or for plants, they would be spores that are produced. And each one, if you look at that telophase picture in the middle, you can see that each one has one of the big chromosome and one of the little chromosomes. So they do not have a maternal and paternal of chromosome 1 and chromosome 2 in each of the cells. They've got one big one and one little one. And in some of those cells, the big ones are blue, And in some of those cells, the big ones are red. In other words, in some of those cells, the big ones are the paternal chromosome, and in some of those cells, the big ones are the maternal chromosome. And then same thing for the smaller ones too. So there are several ways that meiosis produces genetic variation. And remember that we've got our two successive divisions of meiosis that get us 4 haploid nuclei, and each of them has a different combination of genes. And there are a couple of sources for this. First, when they were lining up in metaphase 1 as tetrads, and so we had both maternal and paternal of chromosome 1 all lined up there. There's no reason that maternal needs to be on the left and paternal needs to be on the right, or vice versa. And so you could sometimes, maybe you have, for chromosome 1, you've got maternal on the left. For chromosome 2, you've got maternal on the left. For chromosome 3, you've got paternal on the left. For chromosome 4, you've got maternal on the left. And maybe it's that way this time, and then maybe the next time it'll be maternal on the left, and then paternal on the left, maternal on the left, paternal on the left. Just like flipping a coin, every time you flip that coin, it's an independent event. You can get a heads or you can get a tails, right? You're always going to get one or the other. But just because you got a heads this time doesn't mean you're going to get a heads the next time. And so, even if I've got 3 different chromosomes, that's like 3 opportunities to flip a coin. Half the time, I'll get heads on each of those flips. And so similarly, half of the time I'll have maternal line up on the left. And so if I have a 1/2 chance of getting a heads on the first time, and then let's say I've got a 1/2 chance of getting a heads on the second time, then the likelihood of me getting heads and heads is 1 out of 4. Just like the likelihood of me having maternal on the left and maternal on the left again is 1 out of 4. If, let's say I'm going to flip that coin 3 times, now I'm looking at 1/2 times 1/2 times 1/2. So now I'm at 1/8. I've got 8 different ways that I could go and have these things line up. And so we've got a lot of variety just from those homologous pairs lining up in a particular fashion during metaphase 1. Then we haven't looked at it specifically yet. We will next, but during prophase 1 and metaphase 1, we can have crossing over, and that's where DNA segments between maternal and paternal homologs are kind of mixed. They're crossed over. As you can imagine, we no longer then have a purely paternal chromosome. Instead, we've got one that's maybe mostly paternal with a little maternal. And so you can get this mixing. Obviously, that's going to produce some variation. And then, in the end, remember that your sister chromatids are going to need to pull apart. And so your maternal and paternal chromosomes of homologous pairs, they're going to separate independently at anaphase 1. So crossing over can be a big source of genetic variation, and let's take a closer look at it. And so here you can see a homologous pair. We've got a tetrad lined up. And what happens is if you look at those top 2 strands, let's assume that red is maternal and blue is paternal. That top strand, if you follow that one along, it looks like originally we're kind of going along, and then it went a little bit behind its homologous pair, and then came down towards the bottom. But then at the very end, you can see where we had a chiasmata, we wound up having a little crossing over event between the tail of that blue strand and the tail of that red strand. Now, both of these, when we have these crossing over events, they need to occur in kind of matched areas. So we shouldn't have just like a deletion of genetic information or a repeating of particular information in various strands. And so they occur at the same location. So we're still, let's say that last gene was about eye color. That's still present in each one, but now that top strand that was mostly maternal at the very end has a little bit of paternal information instead. And same thing with that strand that was mostly paternal, now has just a little bit of maternal information at the end. It's no longer purely a maternal chromosome or a paternal chromosome. It's a mix. And we can also see that occurring with those middle strands. And so we have 2 chiasmata that are the result of separate crossing over events. And again, when we have these homologous pairs, this can only occur between homologous pairs. And so chromosome 1 and chromosome 2, they're not having crossing over events with each other. This is only going to happen between the maternal and paternal chromosome 1 or the maternal and paternal chromosome 5. It's always going to be that homologous pair, the maternal version and the paternal version, just kind of swapping some of the ideas about how they can go. So here you can get some more images of this, the synaptonomal complex. So synapsing homologous chromosomes in meiotic prophase 1 are held together by a synaptonomal complex composed mainly of proteins. These synapsing homologous chromosomes, they wind up having these proteins kind of hold them together, and it allows for this crossing over event to occur. So a quick comparison of mitosis and meiosis, we wind up getting different outcomes for this. In mitosis, there is a single nuclear division that produces two daughter cells that are genetically identical to each other and the original cell. And so we start with a 2N cell, and we wind up getting two 2N cells, and they should be identical to each other and the original cell. And again, all of this is assuming that no accidental like little typos or just errors like that. The goal is to get two identical cells. Now in meiosis, we start out with a single diploid cell, and it undergoes genetic rearrangement and two successive nuclear divisions without further replication, and this results in four genetically different haploid daughter cells. And so each of those four cells is genetically different from one another. And then on top of that, we wind up having four of them, and they are haploid. So let's wrap up by looking at some animal life cycles, or just some life cycles in general. In animals, somatic cells are diploid, and they multiply by mitosis. The only haploid cells that are produced are gametes. Gametes develop when germline cells undergo meiosis. We can have gametogenesis, which is going to be the formation of gametes, and then specifically there's spermatogenesis, which forms four haploid sperm cells for each cell that enters meiosis, or oogenesis, forming one egg for every cell that enters meiosis, plus some polar bodies. There's a little bit of difference what happens in the ovaries. And so here you can see that each species, again, has a characteristic number of chromosomes that does not change because the doubling of chromosomes that occurs during fertilization is compensated for by the reduction in the chromosome number that occurs during meiosis. You can see a color code here in a design that's used throughout the rest of the book. For example, in all life cycles, haploid N is going to be shown in purple, and the diploid 2N generation is gold. And so the processes of meiosis and fertilization always link the diploid and haploid generations. Although the actual process of mitosis in fungi is the same as in animal cells, the life cycle structure is a little bit different here for most fungi and many protists. So you can see here that these guys actually spend most of their time as unicellular or multicellular haploid organisms, so N. So they underwent meiosis, and then they can still undergo mitosis after doing so and produce their gametes. Then those gametes can undergo fertilization. We get a zygote, that's 2N, but then pretty quickly they undergo meiosis again. And so most fungi and many protists spend most of their time as haploid organisms. And then finally plants. Plants have a complicated life cycle with alternation of generations. And so you can see it is split, but it's still, again, a little bit odd. Some algae and some fungi also fall into this category. So cells of a multicellular 2N sporophyte wind up undergoing meiosis to form haploid spores, each of which may then divide mitotically to produce a multicellular haploid gametophyte. Then the gametophytes produce female and male gametes by mitosis. The egg and sperm fuse to form 2N zygote that divides mitotically to form a multicellular sporophyte. Notice that we spent most of our time with meiosis talking about the animal cycle, and that's what we're going to focus on here. It's important to know that there are other life cycles. It's also important to know that we have a diploid number that we're looking at, and that there are also haploid numbers, and that plants, fungi, and protists, there can be some variation on the exact formation of the life cycle, but going forward, when you're asked questions on the exam, we're going to be referring to that animal cycle like 99.9% of the time. Thanks, and that's all for Chapter 10.